Good morning, everyone. In the last class, we were discussing about different types of classification system. We had two kingdom classification system, which was inspired by Aristotle's classification attempt. Then we had two kingdom classification given by Carolus Linnaeus. Then we had three kingdom classification system given by Ernst Haeckel, four kingdom classification given by Julius Copland, five kingdom classification given by R.H. Whittaker, and six kingdom classification in the three domain system given by Carl Woese. Later on, we did talk about taxonomical hierarchy as well, where we started from the domain, then kingdom, then phylum and division, then subphylum, superclass, class, order, family, genus, species, then subspecies. So today we are going to talk about the Archaebacteria that is a part of Monera in five kingdom classification, but in the six kingdom classification system, Archaebacteria is a different kingdom. So let's talk about Archaebacteria. Archaebacteria, in as we all know, in Monera or in the normal eubacteria or the true bacteria, the peptidoglycan is the cell wall material of the bacteria. But in Archaebacteria, peptidoglycan cell wall is absent. Instead, a component called pseudomurine is present, which form the primary cell wall component. Then we are going to talk about different types of Archaebacteria. One of them is methanogens. Methanogens are those bacteria that are present inside the component called rumen in the of the ruminants animals or the herbivorous animals such as cows and buffaloes. Rumen is the first chamber of the stomach in the herbivorous animals. So these methanogens reside inside the rumen of the herbivorous animals. There are a lot of more details that it helps in digestion of cellulose, and as it possesses the you can say the enzyme cellulase. Methanogens are strictly anaerobes, autotrophic organism. They occur in marshy areas and convert formic acid and CO2 into methane. So example of these are Methanobacterium, Methanococcus, and one of the you can say theory or you can say fact as well is that when we are talking about greenhouse gases, CH4 methane, methane is one of the greenhouse gas that is causing the greenhouse effect or the global warming. So one of the theories that that apart from all the you can say the pollutants such as sulfur SO2 that is being released from from the you can say car emissions, and the CO2 and the carbon monoxide, apart from that the CH4 that is being released from the rumen of the herbivorous animals. The methanobacterium and the methanococcus bacteria, these are one of the leading cause of release of CH4 methane, and which is leading to a release of this greenhouse gas and causing global warming. So when you think about all the causes of global warming, one of the reason is the release of methane gas from the rumen of the herbivorous animals. It is one of the contributor as well. I particularly do not know the particular statistic as to what exactly is the percentage which is the contribution of these methanogens, but it is one of the part which does release methane gas. So one of the another category that we have is halophiles. Halophiles occur in the salt-rich areas such as marshes. Example of these are Halobacterium and Halococcus. These are aerobic chemoautotrophs. What do we mean by chemoautotroph? It means it requires chemicals to synthesize food and energy. They can synthesize their own food and derive energy from it, but they require chemicals for that. So they have red carotenoid pigment called bacteriorubin, which protect them against harmful solar radiations. Halophiles can live under saline condition because they have special kind of lipids in their cell membrane, occurrence of mucilage covering, high internal salt content. So if we have to talk about why exactly halophiles can survive inside, you can say, a very salty area. I'll give you one example for it. If we have this, let's say, a bacteria in a very, you can say, salty area, salty water. There are, let's say, few salt molecules inside the bacteria and a lot of salt outside. So according to osmosis, the water content will go from the high water content to the less water content. So the water will go from here, and this will lead to this bacteria get flaccid. So what exactly is happening with halophiles is that this bacteria is having a lot of internal salt content already inside it. So when there is salt content outside, there is no what, you can say, it is matching the almost the similar amount of salt content inside and the same amount of salt content outside. So there is no, you can say, overall water exchange. But when we are saying there, let's say if there is still salt content exceeds the inside when it as comparison to outside, then water will try to come outside or inside. It is not really possible because there is membrane of pseudomurine and also the selectively permeable plasma membrane which does not allow free exchange of water. So this exchange is not possible. It is prevented by plasma membrane and also the cell wall. So the halophiles can survive in the extreme salty area as well. Halophiles give off offensive smell as they can decompose organic matter as well. So one of the other category that we have is thermoacidophiles. Thermoacidophiles get their name from because they can survive high temperature as well as high acidity. When we are talking about high acidity will mean lower pH. So they live in hot sulfur springs where the temperature can reach up to 80 degrees Celsius and the pH can go down till pH 2. Example of these are thermoplasma and thermoproteus. Under aerobic condition, they can oxidize sulfur into sulfuric acid. You can see the reaction here, 2 sulfur plus 2 H2O plus 3 O2. We get 2 H2SO4. In anaerobic condition, the thermoacidophile reduce sulfur to H2S when the oxygen is lacking. So instead we get H2S. Thermoacidophiles are able to tolerate high temperature and high acidity because they have the branch chain lipids in the cell membrane. So when we are talking about branch chain lipid, what do we exactly mean? The lipid structure is like this of the plasma membrane. So these are, you can say, uh, like this. The lipids are like this of the regular, you can say, a bacteria. But a branch chain lipids will be in up nearby. There will be branching between the lipids. So this will help in providing a stronger bond to the lipid molecules, and it helps in providing resistance to the higher temperature and the lower pH. So present, and also they have special resistance enzymes capable of operating under acidic conditions. One of the example that I will give you regarding thermoacidophiles, uh, one of the bacteria that you are going to get to know in the next year or the in the chapter of biotechnology, it is bacteria name is thermus aquaticus. This is one of the bacteria from which we derive one of the enzyme that helps in the process of PCR. PCR, or you can say the RT-PCR. RT-PCR is one of the tests that we used to do for COVID testing or a lot of viral viral, you can say pathogen testing. So RT-PCR is helps in diagnosing a lot of viral and bacterial pathogens in present inside the body. So when we are talking about you can say three categories that we have: methanogens, halophiles, thermoacidophiles. We have one fourth category, but it is not really talked about. It is called as psychrophiles. Psychrophiles are those bacteria that are able to survive in lower temperature, or you can say freezing temperature. But why we do not talk about psychrophiles exactly in Archaebacteria? Because psychrophiles can exist in eubacteria as well. So psychrophiles are not only limited to just Archaebacteria, they exist in eubacteria too, as they can survive in lower temperatures. We have some more general points about Archaebacteria. This is one of the important point. We are going to learn about this gram staining later on when we are going to talk about eubacteria. Gram staining, it is a process which that we utilize in order to you can say classify bacteria into two categories. Let's say I'll just give you a brief hint. Gram staining divides the eubacteria into gram positive and gram negative. Gram staining is based upon the phenomena where when we have a thicker peptidoglycan layer in gram positive bacteria and we have thinner peptidoglycan layer in gram negative peptidoglycan. So when we apply gram stain on this you can say gram positive bacteria, we get a purple color bacteria. But when we apply gram stain on gram negative bacteria, we get a red, or you can say pink bacteria. Why exactly? Because there is more technicality to this. We have a secondary stain called safranin that we add, which imparts color to this gram negative bacteria, but is not able to impart the color to gram positive bacteria. So purple color bacteria is visible in the gram positive. But why exactly am I talking about gram staining when we are talking about Archaebacteria? Because gram staining is not used to classify Archaebacteria, because they have varying response to the gram stain, as Archaebacteria lack peptidoglycan in their cell wall, which make them very similar to gram negative bacteria. So even though they are similar to gram negative bacteria, we do not really recommend using the gram staining to categorize Archaebacteria. So that's all for today for Archaebacteria. Because I know this is a very small video, because Archaebacteria, even though it does have a lot of details, but those details are not really required for you can say when it comes to studying about the syllabus of Neet. So that's all for today. Thank you.